I'm Boris. I'm a PhD student at the Biomedical Informatics group at Oslo University. And I'm going to talk about the GSuite hyperbrowser, something that we are developing in the group. What is it? Well, we would like to say it is a comprehensive solution for integrative analysis of track collections, aka something that we call GSuites across the genome and the epigenome. It is built on top of the Genomic hyperbrowser, which is a system for statistical analysis of genomic datasets. It has been extensively tested by its users and its developers so far, so we build on these core functionalities, and it's integrated with Galaxy, which allows us to have reproducibility, the user administration, and dataset management. Why would we actually build a system working with collections? Well, one of the reasons is recent large-scale undertakings that generate huge amounts of data, like ENCODE and roadmap epigenomics, which represent a variety of functional elements across a large number of cell types. So an analysis taking all of this into context, we think it's a natural next step for us. And for this reason, we developed the GSuite format, which is used for formal representation of dataset collections. When we developed it, we kept in mind to make it simple and intuitive. So it's a tabular format where each line is URL referring to the constituent track or the dataset. And additionally, to facilitate statistical analysis, it would contain metadata for each dataset and metadata summary for the collection as a whole. You can see more about the specification at this link. As part of the system, first step would be to compile such a collection, so to locate the datasets, put them all together in a single GSuite. We can use public repositories to find these datasets, which in general are not designed for the extraction of large number of datasets together according to some shared characteristics. So getting all transcription factors for a cell is not an easy process usually in these repositories. To solve this, we rely on the concept of database views. So we develop tools that allow you to browse and select remotely located tracks based on some characteristics. We provide two access points, a low-level access point that just gives you all metadata that is found in the repository, which you can browse, and it's literally a copy of the metadata found in these repositories. We do this by crawling the public databases. But this way of using it might be a bit overwhelming because there is a lot of metadata there. You have to be like an experienced user to understand all the attributes. So we also provide a high-level access point, which is kind of a curated catalog of all these databases that we support. And the currently supported repositories for browsing are ENCODE, roadmap epigenomics, ICGC, TCGA, and National Human Genome Research Institute GWAS catalog. We also plan to support Blueprint and Phantom 5, and we would gladly accept any requests for other repositories. Another way to compile GSuites would be from datasets in your history. something similar that you can do with Galaxy dataset collections, or from an uploaded archive, a zip or tar archive that you upload to the server, and that can be then extracted to a GSuite. Or also a useful case would be creating a collection of datasets from a single dataset according to some categorical value inside. So an intermediate step in an analysis, in a usual analysis, would be also to customize the GSuites. I would like to say that here that we also differentiate between three types of GSuites, three types of collections. Remote GSuite will contain references to datasets that are found somewhere else, usually in the repository that you've searched through. After you download the datasets, the GSuite is, we call it primary, meaning that it's in a primary format that the datasets are in at the repository. And to use them in an analysis tool at the GSuite hyperbrowser, we pre-process them into a binary format that's more convenient for analysis and, of course, more efficient. So these are just some of the tools that we offer to customize the GSuites. Of course, selecting subset of the GSuite, subset of the metadata columns, edit some metadata, modify each track in each dataset in a collection in the same way. Of course, if you need, you can always export them in history and have them as separate datasets. For the statistical analysis of collections, first I would like to mention that the whole system is developed as a big collaboration with a lot of biologists and statisticians, and with their input we have created 5 generic statistical questions that can be asked about collections of genomic datasets, beginning with asking which tracks in a suite are most representative and most typical for the suite. A useful example would be to, let's say, find an outlier in your of replicates of experimental tracks. Another question that you can ask is which tracks in a suite coincide or co-locate most strongly with a separate query track. A useful example would be comparing the enrichment of a set of trait-associated SNPs to, let's say, a collection of open chromatin regions datasets for different tissues. It would give you a chance to do some comparative analysis of it and see how enriched the query track is in the reference tracks in the collection. When having 2 GSuites or 2 collections of datasets, you can ask the question: are certain tracks of one suite coinciding particularly strongly with certain tracks of another suite? Creating a heat map as an output. A useful biological example would be to assess the enrichments of somatic variants of different cancer types in heterochromatin datasets for different cell types. And the last 2 questions are in regard to different regions of the genome. So you might want to ask in which regions of the genome the tracks of a suite have the most occurrences, like trying to find genes with particularly high frequency of somatic variants across a set of cancer patients. So it might be interesting to find out which somatic variants are occurring together along the genome. in a way. And the last generic statistical question that we offer is in which regions of the genome do tracks of a suit exhibit the strongest tendency to co-occur. An example related to this could be finding the regions of the genome where ChIP-seq peaks for transcription factor binding sites co-occur frequently together in a way. So for all of this, we developed some tools, but we also put in some effort of usability of the system. For new users, we offer a basic operation mode, which can be used to quick start the users of the system. And for this, we'll offer a catalog of predefined biological questions. The analysis is guided, the analysis workflow is guided, and usually in the tools you will use default parameter values, and it's less overwhelming to use it. We will see a demo of how easy it is to start with the system in just a few steps. And of course, if you are a more experienced user, or after some time, you can use the advanced operation mode where you get full access to the analysis parameters and get more power in your analysis in a way. So I've had bad luck before with live demos, so I prepared a screenshot this time. So I don't care if there is no internet connection or anything. It's not working. Play this. So you can find the system on hyperbrowser.uio.no/gsuite. This is the front page where you can see, can get some explanation and also choose, let's say, the basic mode. If you're starting with the system, we offer 3 groups of biological questions to get you started. Let's say that you want to ask if some specified trait-associated variants are particularly active in a certain cell type. We give you a full analysis steps instructions here. In this case, you need to go through 3 steps. Supply the traits. In this case, we supply a demo track, which is related to multiple sclerosis associated regions. Expanded 10, sorry about that. That didn't work as planned, but okay. So the query track that we supply is multiple sclerosis associated regions, expanded 10 kilobases on both sides, in both directions, and to supply a GSuite or a collection of data sets. In this case, you can use the curated search tool. And to see how easy it is, we just get 5 categories here: open chromatin, and let's say we use DNA's hypersensitivity size as a marker for open chromatin regions, and it takes a little time. I don't because it's searching the database, I guess. And it will give you, as you can see, results from both ENCODE and roadmap epigenomics. For this case, let's use the roadmap epigenomics that repository and broad-peaks type of data sets. You can see there are 78 data sets. You just click execute, and it takes, in this case, it's pretty quick because we are also caching this kind of collection data sets. And now you have your data sets in your history. The second element is the GSuite representing them. And to run the analysis, the actual analysis, we can use the to which tracks in a suite coincide most strongly with a separate single track. We select the query track, the multiple sclerosis track, and then the collection of DNA hypersensitivity sites there. And yeah, this is displaying all the metadata that is available for these datasets in the database. We select the start and epigenome name as an important information that we want to see in the results. I click execute, and this takes around a minute, so I just pause here, and which is, I would say, quite fast for around 80 analyses. And you get the results. That track is actually mostly related to multiple sclerosis. We can see that there are immune cells on the top and fetal brain, like with the lowest association. We can see the similarity scores, and by clicking these checkboxes, you can actually generate some charts also, showing which tracks in your collection are actually most related to the multiple sclerosis track. So this is just a simple example, but you can do this with any datasets that. Of course, integrating with Galaxy dataset collections is something we are planning. We were planning it for a long time, but one thing that we were missing from Galaxy dataset collections is the metadata, which is quite useful in statistical analysis. So yeah, that's something we will try to figure out later. I think that's about all to say. If you have any questions.
I had one question. I'd seen you mentioned the 5 statistical questions that you'd prepared. What if we had a 6th statistical question?
You can always contact us and figure out the solution.
Okay, that's the answer I was hoping for. Thank you. Any other questions? Thank you.